Okay, so thank you everyone for joining. As I said, my name is Donna. I am with the Lake Hopatcong Foundation and I am the Grant and Program Director. For those of you not familiar with our foundation was founded in 2012 and our mission is to dedicate ourselves to protecting the lake environment and enhancing the lake experience, bringing together public and private resources to encourage a culture of sustainability, stewardship, on and around New Jersey's largest lake for this and future generations. Our mission is very far-reaching, and we do work in the environment, education, community building, public safety, recreation, as well as arts and cultures. I just want to take a moment to introduce Dr. Fred Lubnow. He's the Director of Aquatic Programs at Princeton Hydro. His areas of expertise include aquatic and watershed management, restoration ecology, and identification and management of hazardous algal blooms, what we refer to as HABs. He's involved in a variety of lake and watershed restoration projects, including some of which he's been working on at Lake Hopatcong since, I would say, the early 2000s. So thank you, Fred, for joining us. If you'd like to take a moment to share your screen, we can turn the program over to you.
All right, sounds good. Okay. All right, so can everyone see that?
Yes.
All right, excellent. Well, thank you for inviting me to give this presentation. As Donna mentioned, I've been working on Lake Hopatcong for a number of years. I've been working since the early 2000s when the commission was first formed. And I actually worked in the 90s too, when it was the regional planning board. So it's funny, a lot of the data I'm going to show, the 30-year data set sort of spans my professional career as an environmental consultant. So it's sort of funny that way. But the presentation is called Lake Hopatcong Water Quality Monitoring Results, a look back and what's happening in 2020. So, all right. So the first thing I usually, when I'm talking about monitoring, I like to ask the question, well, why even conduct water quality monitoring? And actually with the Lake Hopatcong community, this is a question that I usually don't have to go into too much detail, but I did want to quickly review this. Why we do water quality monitoring? Well, first, it's a checkup on the lake. It's identifying the overall health of the lake. So it's like going to the doctors to get a checkup. Another reason why we monitor is to see if we're in compliance with state water quality regulations, and I'll touch a little bit on that through the presentation. Something that's really important is identification of potential invasive species. So while we didn't identify the water chestnut, the foundation and their volunteers identified water chestnut, which is a nasty aquatic plant that's an invasive species, and thankfully their efforts of identifying that plant and physically removing it means it's a lot more easy to manage, and it's a lot lower in cost than dealing with pesticides and large-scale harvesting for that particular plant. Another reason why we like to do water quality monitoring is to identify long-term trends or changes. I've been to many a public meeting where someone will get up and say, when I was a kid, I used to be able to see my toes in the water and now I can't. And that's meaningful information. But if that same individual says, well, I have a graph of water clarity over the last 30 years and it's been on the decline, having that data makes that argument a lot more, it makes it stronger, particularly if you're trying to convince someone to provide funding for a project. We also want to determine the relative effectiveness of any in-lake or watershed management measure. So I'll be talking about some of the projects we're doing with the HAB grant. And as part of those projects, we're collecting water quality data to determine the cost effectiveness of those techniques. And then finally, another reason why we collect water quality data is that information is really important when you're seeking out funding for implementation projects. So state, federal, county, local agencies really like to see that water quality data is being collected even before you're asking for the funds, so that you can identify a problem and you have a way of quantifying, if you're going to implement a project, how effective it is And even if it's not effective, very frequently that data can be used to modify the implementation project so it is effective. So that data is really useful in terms of identifying whether or not a project is working, and if it's not working, it helps to identify how to fix that situation. So real briefly, Lake Hopatcong's water quality monitoring program, there are 11 standard long-term monitoring stations that we sample 5 times over the course of a year, once a month between May and September. We collect in situ data. In situ essentially means we're collecting the data right there on the lake, so it's with a meter. And we'll measure temperature, dissolved oxygen, pH, and conductivity from surface to bottom at all 11 stations. We also measure water clarity with a Secchi disc, and that's that little black and white disc you can see in the lower right. It's very useful information. It gives us an idea of how clear the water is. It can also identify whether or not there's changes seasonally or changes from year to year due to algal blooms. We also collect discrete samples for phosphorus, nitrogen, ammonia, total suspended solids, chlorophyll A, which is a photosynthetic pigment that all algae use, so it's a way of measuring the amount of algae in the water. We also collect samples for phytoplankton, which are the free-floating algae, the zooplankton, which are the little micro animals, some of which directly feed on the algae. And then we collect observations on the submerged aquatic vegetation, the weeds. The monitoring program started in the early 80s with the original Clean Lakes Phase 1 diagnostic feasibility study, which was funded through EPA. But this monitoring has been consistent since 1990. So you essentially have a 30-year data set for Lake Hopatcong. It's probably one of the longest consistent databases. I know it is in terms of aquatics for New Jersey, and it may be for the Mid-Atlantic. So it's rare to have such a large database, which makes it very useful, and I'll go into that as we go through the presentation. And then finally, we started in 2016 based on recommendations by the Commission and Foundation. We've been collecting nearshore samples for cyanobacteria cell counts and cyanotoxins in July and August. And we did do that. Notice that started even before the HAB incident of 2019. So there was already concern over these cyanotoxins. So the next slide is just showing you a map of the lake and showing you these standard monitoring stations, which are in red. The yellow stations were ones associated with grants that have been completed. And with the HAB grant, we have additional stations. So the red dots show you where we've been collecting data for about 30 years. Other stations we add and will remove based on what sort of projects are being implemented. So I'm showing this slide here. This is showing you dissolved oxygen concentrations from surface to bottom in the middle of the lake, where it's about 46, 50 feet deep. So if you're looking vertically, those numbers are in meters. And then you're seeing across the top the dissolved oxygen concentrations. A Lake Hopatcong is what's called a dimictic lake, which means it typically mixes twice a year. It mixes in the spring after ice out, and then it will mix in the fall after things start to cool down. So during the summer, you have what's called thermal stratification. And if you've ever jumped into the lake and the surface feels nice and warm, if you dive a little deeper, it gets really cold. That's because as you go through the water column, there'll be a quick drop in temperature. That drop in temperature is called the thermocline. And so it seals the bottom off from the surface. The result of that is what you're seeing in this slide, that you have dissolved oxygen concentrations at the surface being sufficient to support a healthy fishery and lake community. So you can see that the dissolved oxygen concentrations at the surface are above 9 milligrams per liter. But once you hit that change in temperature, there's a quick drop in dissolved oxygen. And so when you go down to the deep waters, you can see there's very little dissolved oxygen at the bottom. And there's a number of reasons for that. One is because of that change in temperature, the lake isn't fully mixing during the summer. So the bottom waters can't be replenished with oxygen from the atmosphere. You only have bacterial decomposition going on in those deeper waters, so that removes oxygen. and it's dark down there, so there's no photosynthesis, no plant or algae growing. And in photosynthesis, that produces oxygen. So this is very typical of a deep dimictic lake, that during the summer months, you have very little oxygen at the bottom. So not only does that mean you have no fish life deep down during the summer months, but also there's a chemical reaction from the sediments that phosphorus can be liberated from the sediments. That's called internal loading, and I'll touch on that as we go through the presentation. But I did want to show this, that this is very typical of a deep dimictic lake like Lake Hopatcong, well oxygenated during the summer months at the surface, but very little oxygen in the bottom. I'm going to shift gears, so that's just one profile of dissolved oxygen, but this is something that's really telling. This is basically a graph of surface water temperatures at that same mid-lake station for July. And I did this last year because we were talking about the HABs and we were dealing with the HABs. So I wanted to see if we can see the impact of climate change on Lake Hopatcong. And I was surprised how strong this came out. This is a statistically significant relationship. Over the years, we've seen this increase in temperature. Now, there's variability from year to year. For example, 2018 was a relatively cool year, and you can see that dot is lower. But 2019 and this year's data point was very high. For the last 30 years, we've seen this increase in surface water temperatures at Lake Hopatcong. So again, this is showing you that the impacts of climate change. And just to put this into context, if you think of, like, if you look at 25 degrees Celsius, so I'm picking that just because that's between the optimal and the tolerant level for brown trout. So in the 90s, the July temperature exceeded 25 degrees Celsius twice in the 90s. In the 2000s, it exceeded 25 degrees Celsius 3 times. The last decade, from 2010 to 2020, every July temperature was above that 25 degrees threshold. So we have seen this steady increase in temperature at Lake Hopatcong. The reason why that's important is what really triggers a cyanobacteria algal bloom, or a HAB, are 3 conditions. High seasonal water temperatures. Most of the cyanobacteria like warm water. The second component is they like those still water conditions. They like thermal stratification, and I'll explain why in a minute. And then third is they need a lot of phosphorus to grow. So concentrations as low as 0.03 milligrams per liter can generate nuisance scums or blooms. So you need a lot of phosphorus, still water conditions, and high water temperatures, and that will typically trigger a cyanobacteria algal bloom, or a HAB. Now, the cyanobacteria are pretty unique in that technically they're not algae. They're actually bacteria that pretend to be algae. So they're bacteria that can photosynthesize like algae. And I'm showing these 3 slides to show you how unique these cyanobacteria are. So if you look at the slide in the upper left, that's one filament of cells of cyanobacteria. And that big, large cell in the middle, that's called an aconite. So what happens is when you hit fall and things start to cool down, those aconites will settle to the bottom. They're resting spores. They'll stay there, they'll overwinter, and then when the lake mixes over in the spring, brings those aconites to the surface, they hatch, and that's where you get your cyanobacteria. The slide still over to the left, and the lower one, again, you see another filament of blue-green algae, and if you look in the upper right, you see that clear cell. That's called a heterocyst. And many cyanobacteria can fix their own nitrogen. And this is why we focus so much on phosphorus, because to fix nitrogen, it's very expensive from a physiological standpoint. So they need a lot of phosphorus to do that. So in freshwater environments, we're almost exclusively focusing on phosphorus. Nitrogen is important for algal growth, but with phosphorus, when you increase phosphorus, not only are you increasing overall algae, but you're increasing more of the bad algae, the cyanobacteria. If you're dealing with the shore, like marine or estuarine environments, nitrogen is a lot more important for those systems. But with freshwater systems, phosphorus is generally the primary limiting nutrient. That's why we focus so much on controlling phosphorus going into Lake Hopatcong. And then the last thing I want to show, that photo to the right is showing you a sample of algae, and you can see that graduated cylinder, and look at the concentration on the surface. Those are cyanobacteria that are using their gas vacuoles. So the thing about the cyanobacteria is they can float to the surface where they outshade the other algae. They make those surface scums, but then they can fuel up on all the sunlight they want. And then they can collapse those gas vacuoles and sink down to deeper waters where there's more nutrients and fuel up on nutrients. So they're pretty ingenious that they can move up and down the water column, but they like those still water habitats to do that. So if you have a system that's very well mixed, it makes it more difficult for them to use those gas vacuoles and create those surface scums. So I just wanted to give you some unique features of the cyanobacteria as we go into the data. So I'm going to start talking about some of the data. I'm really going to focus on this year and last year. So last year was an unusual year in that we saw more phosphorus early in the season than we typically do. So look at the red bar across at that 0.03 milligrams per liter. I can tell you that for Lake Hopatcong, we want our average, generally our average concentration of phosphorus in the lake to be 0.03 or lower. Generally, that will control the amount of algae in the water. So last year in May, you can see that we had higher concentrations of phosphorus in Lake Hopatcong compared to this year. So this is just showing you the phosphorus concentrations for all of the stations that we monitor long term. So more phosphorus in May last year than this year. Similar, if you look at June, now I can tell you that our June samples we collected last year was less than a week before the blooms hit the lake. So it was unique in that we actually had data that we collected less than a week before the bloom really triggered. So even when we had that data, we saw that we did have higher phosphorus concentrations in the lake in June compared to what we've been seeing this year. So I can tell you that this year is more of a typical year at Lake Hopatcong, which I'll get into in just a second. But then lastly, I want to show you July. So this is data we just collected a few weeks ago. In July of last year, the phosphorus concentration started to go down, but we did see this year higher phosphorus concentrations in July, which is typical. Usually when the blooms hit Lake Hopatcong, it tends to be toward the end of the growing season, and frequently it's after Labor Day. So once it's after Labor Day, it's not as much of an issue for a lot of people, since in a normal year, the kids would be in school, people are back to work, the beaches aren't open. So even though you do tend to have these halves, they tend to happen in September or October. What was unusual about last year is the blooms started at the beginning of the growing season. So this graph is showing you, I graphed the long-term average concentration of total phosphorus in June for the last 30 years. And what's really telling is when you look at, what's really telling is when you look at 2019, it is one of the highest June concentrations. It exceeded 0.04, and we haven't seen that in quite some time. So that's what really triggered those blooms that happened starting in June of last year. It was, we had unusually high phosphorus concentrations. And what's really interesting, and I've used this at public meetings, where you can see before 2000, when it was the regional planning board, you know, they did what they could with the limited amount of funds they had. They did the harvesting. They had very little money to implement projects. And you can see back in the 90s, we had some pretty high phosphorus concentrations. Once we hit 2000 and the commission was formed, and the commission had money, they did very aggressive harvesting. They had shared services agreements with their staff cleaning out catch basins. We got a few grants. We got $2.5 million in grants to implement a variety of stormwater projects. So all that was going on great. And you can see through the 2000s, we got the concentrations down. And the biggest complaint I remember were weeds, which makes sense. When you have less algae, the water's clearer, sunlight is hitting the sediment, so you get more weeds. So we were battling mostly weeds. And then when the recession hit, a lot of funding dried up. There wasn't a lot of money. By 2015, the grants ended. And you can see that once we hit, like after 2010, this slow increase in phosphorus over the last decade. And that average concentration that went over 0.04, it was almost like, you know, a bell going off going, hey, the lake's heading for some trouble. We need to do something. So this is the value of this data, that it's allowing us to identify these trends, and it allows us to implement planning and projects for the future. Now, what was, again, unusual about 2019 was, so not only was it a warmer year and we had more phosphorus, but this is the reason why we had more phosphorus. So if you look at the rainfall data, what was happening in June, and I can compare this to 2018. 2018 was a pretty wet and cool year. 2019, as we were going into spring, was more of a normal year, but we were getting into this pattern where we'd have these intense storm events followed by a few days of sunny weather. And you can see this, you know, these bars are showing you how much precipitation fell, and then you'd see a few days of nice sunny weather. So you'd get these intense storms that would rinse the watershed, transport all the nutrients into the lake, and then you'd have this nice sunny condition. So this alternating storm, sunny weather really is what triggered these blooms. So bringing the phosphorus in, plenty of sunlight, plenty of phosphorus, and the HABs took off early in the season. That was the result last year. Last year was, you know, I mean, everyone on this presentation, I'm sure, is very familiar with what happened last year at Lake Hopatcong. And it wasn't just Lake Hopatcong, it was other lakes experienced this literally at the same time, and not just in New Jersey, but in New York and Pennsylvania too. We were shocked on how regional it was. And that's why I really attribute it to this weather pattern, because we saw the same thing at Harvey's Lake, a lake we monitor in Pennsylvania. We saw it in a number of lakes we deal with up in New York, like DeForest Lake, Greenwood Lake suffered through this. It was the larger, deep lakes that really suffered through these HABs. And again, if you're a large, deep lake, that water rinses in. That means that water is going to stay there. Your flushing rate is low. It's not like a small, shallow lake that if you have a big storm, it can wash a lot of the nutrients and the algae away. In these larger, deeper lakes, they hold that water, hold those nutrients, and this is the result. But again, I do want to emphasize that we have seen these blooms in the past at Lake Hopatcong, but they tend to happen under a, quote, normal year, unquote, in September and October. So this was when and I'm pretty sure Donna sent this photo to me. This is showing you a bloom in October. So again, October, people aren't recreating as much. The beaches aren't open. So yes, it's problematic, but it didn't have the devastating impact that the HABs did in June, not only on recreation, but the local economy. And matter of fact, later today, I'm on a town hall about the impacts, the economic impacts of HABs, just nationwide. So that's later on today. But this is showing you that these HABs do occur at Lake Hopatcong. And that's why we're moving forward in all these efforts to implement watershed and in-lake projects to avoid or at least minimize the impacts of these HABs. So this is showing you this is just a general, I want to put this into a sense of why we're looking at chlorophyll as a long-term goal for Lake Hopatcong. So I mentioned chlorophyll A. It's a photosynthetic pigment all algae have. The TMDL, so the total maximum daily load, years ago we calculated we have to put Lake Hopatcong on a phosphorus diet. And when we did that about 12 years ago, we've implemented projects that have reduced what we need to get the level down to by about a third. So that's good. It took 12 years to get about 33% achieved, but we still have a way to go. And what we want is we want the average chlorophyll A concentration to be 8 micrograms per liter. So look at the graph up above. You can see if you have a chlorophyll A concentration of less than 10, your cyanotoxins should be, less than 20,000 cells per mil, and then your microcystin, which is a type of cyanotoxin, should be less than 10. So again, this is all sort of links together, that the TMDL is saying we want the chlorophyll A to be at 8, and if we're at 8, we should have no to very few problems relative to cell counts and cyanotoxins. Well, what are cyanotoxins? Well, just real quickly, they're a diverse group of low molecular weight molecules that can really have an impact on the health of people, livestock, and pets. Um, there are various strains within a certain species of cyanobacteria. There's a lot of research being done into what really triggers the production of these cyanotoxins, and the only thing that is the overlying theme is some type of stress on that particular bloom will make them generate these cyanotoxins. And there are three main types. There are hepatotoxins that affect the liver, neurotoxins, and dermatoxins. I do want to emphasize cyanotoxins are not taste and odor compounds. This comes up quite a bit. If cyanotoxins are odorless, colorless, tasteless, so you can't detect them by looking at the water, you can't detect them by smell or by taste. You can only detect them by measuring them. There are taste and odor compounds that the cyanobacteria generate, such as geosmin and MIB, but they do not produce any negative health impact. So the nasty, like earthy or rotten fruit odor you may get from a bloom is because of geosmin or MIB, but unfortunately there's no simple relationship between the taste and odor compounds and the cyanotoxins. We have some lakes we work on, some drinking water supplies we've been working on since 2012 that we never get any cyanotoxins, but they're constantly battling taste and odor compounds. And then on the flip side, there are many times where we're getting a measurement of a cyanotoxin, but there's no taste or odor problem. So again, I want to emphasize that just because you have a nasty taste or odor does not mean you have cyanotoxins. So some of the more common ones, here are the three that New Jersey is concerned about. Microcystins, which is a group of 100 toxin variants. It's the most common toxin that's found in the environment. So there are draft thresholds of concern that both EPA and New Jersey DEP have. There's also one called Cylindrospermopsin. We've been monitoring for this one we've been monitoring for microcystins for about 5 years, and we have gotten measured concentrations from time to time. Cylindrospermopsin, we've been measuring maybe for 3 years, and we may have gotten one. It tends to be very rare. And then there's anatoxin A, which only DEP has a threshold for. And we've been started monitoring anatoxin A this year. We have not found that one yet. So the one, if we find any, it tends to be microcystins. So if you look at the next slide, it's showing you New Jersey's recommended threshold for recreational water bodies compared to EPA's. So they tend to be lower. And you can see New Jersey has a concentration for anatoxin A. EPA is working on their threshold for that particular toxin. So when we're going out measuring, we're looking to see if we're above these concentrations. So I did mention that we have been monitoring since 2016 for cyanobacteria and cyanotoxins. So these monitoring events are very different. So I showed you that figure that showed the lake and those in-lake, mid-cove, mid-deep stations. They've been monitored since the 90s. These stations have been established. They're near the beaches. They're near the shoreline. And we just started this in 2016. So we collect, again, temperature, dissolved oxygen, pH, conductivity. We measure water clarity with a Secchi disc. We measure the amount of cyanobacteria cells, and then we measure for cyanotoxins. So I can tell you that we've done, this year, we've been monitoring for all three of those cyanotoxins, total of 8 stations. So we completed the July event. We have another one coming up in July, and I can tell you that of the 8 stations, we measured all 3 cyanotoxins. All 3 came up negative at all 8 stations. So no measurable amount of any of the 3 cyanotoxins. And even last year, during the high cell counts, we weren't getting any measurable amounts of the cyanotoxins, but the reason why the state agencies focus on cell counts is these are just 3 of dozens upon dozens of compounds of cyanotoxins and other compounds that can have a health impact. So that's why DEP uses cell counts, because there's a higher probability, the higher number of cells, that maybe there's some other compound out there that may have a negative impact on human health or pets. So, you know, we currently are working with DEP to use our data as a guide so DEP can make a determination on whether to actually test or to post action alert levels. So, and that's a conversation that the Commission and Princeton Hydro had with DEP very recently. So we're going to be providing our information to DEP so that they can make decisions on how to proceed with Lake Hopatcong in terms of them going out and testing and possibly putting an action alert level up. So again, I want to emphasize that in freshwater systems, more than likely the harmful algal blooms are the result of cyanobacteria. When you're in a marine or estuarine area, there are other algae such as dinoflagellates that can produce harmful algal blooms. But in freshwater systems, it's almost always cyanobacteria. So I'm going to wrap up the presentation by saying, okay, well, we have this data, we're looking through it. What do we do with it and how can we move forward? So this is something I presented last year during the nasty HAB. We were going through that. Really, there's a three-tiered approach on dealing with HABs. So the one is the global issues associated with climate change. So from a local basis, from a local perspective, we're not going to have that direct and immediate impact on the influence of climate change on the lake. So that's one tier. That's more of a global issue that needs to be addressed. The next issue is a watershed implementation plan to reduce the phosphorus concentrations entering Lake Hopatcong. And we actually developed one back in 2006. And that's where we identified we want the phosphorus to be at 0.03 milligrams per liter or lower. And we want the chlorophyll to be at 8 or lower. And with that plan, like I said, we've implemented we've reduced the targeted load by about a third. That plan needed to be updated, and fortunately, the Commission received a grant through the New Jersey Highlands Council. So we are finalizing the updated WIP. So we updated that plan, and it's important to update these plans between every 5 to 10 years. If you have a lot of development or land use going on in your watershed, probably updating it every 5 years should be done. But if you have a relatively stable watershed, not a lot of large changes in land use going on, updating it maybe once a decade should be fine. And obviously it was beyond a decade, but we did just very recently update the watershed implementation plan. So that's identifying projects throughout the watershed, stormwater, septic management, green infrastructure, shoreline stabilization, stream bank stabilization, projects in the watershed that can really reduce the phosphorus going into the lake. But then the third tier is we have to look for options for these localized bays, coves, beach areas. What can be done on a very local basis to deal with a HAB on a small scale basis? And that's really what the HAB grant that was awarded to the Lake Hopatcong Commission is really focusing on. So I just wanted to give you a perspective. So here's Lake Hopatcong. That dark blue line is showing you the watershed. So rain hits anywhere in that land. You can think of the watershed as a big funnel. It rinses everything and eventually goes into Lake Hopatcong. So I just want to talk, I mentioned the HAB grants. So the Commission was awarded $500,000, and I'll break that down in a little more detail in terms of additional funding that was provided for it. But we're implementing a variety of projects. And again, these are focusing not on large full lake things, but on small scale nearshore projects. So we're implementing projects in Ashley Cove. We just started the monitoring. We treated with a strong oxidizer because there was quite a bit of filamentous mat algae in there we had to knock down, and we're going to start treating with Phoslock. We've also installed biochar, and we've refurbished a floating wetland. We're going to refurbish a floating wetland island and install some new ones. So we have a number of projects we're going to focus on in Ashley Cove. In June, we conducted a large-scale Phoslock treatment. Phoslock is a clay-based product that latches onto phosphorus, making it unavailable for algae growth. It's sort of like alum. If you're familiar with alum, alum does the same thing, but alum has aluminum in it. Phoslock does not have aluminum in it. So that's a good thing with Phoslock. Another good thing with Phoslock is it will bind with organic or inorganic phosphorus, where the alum will only bind with inorganic phosphorus. The disadvantage with Phoslock is it's a lot more expensive. So we did do a large-scale treatment. It was one of the largest in the United States, a 50-acre area in the landing channel area of the lake. We've used Phoslock in other lakes, but we tend to use it in very small areas because it is expensive compared to alum. But we did collect some post-treatment data, and the results are very promising. We're working with the municipalities and Beach Club to get 3 different types of aeration systems installed. So these aren't full lake aeration. These are shoreline beach areas. So one will be an air curtain. Again, remember the blue-green algae like those still water habitats. So if you can agitate the water, it makes it more difficult for them to make the blooms, and it makes it more difficult for the cells to accumulate along a shoreline. And then we're installing some pretty new technology called nanobubbles. This is something that's new to us, so we're installing 2 systems. One will be a nanobubble system onto itself. The other will be a nanobubble system with ozone. And the ozone helps to break up and destroy the algae, and it's also supposed to break up the cyanotoxins and taste and odor compounds. So we're literally, we'll be sending an invoice next week to the commission so we can purchase all the equipment. We're hoping to install it within the next 1 to 2 weeks, these 3 different systems that we'll be evaluating. Another thing that we're doing is we're going to evaluate an alternative algicide. So 9 times out of 10, the algicide that's used to control algae is copper sulfate. And copper is problematic in that it can kill non-target organisms. It settles in the sediment. If you have high enough copper concentrations, it's expensive to dispose of that material if you want to dredge. The advantage to Green Clean is it's a strong oxidizer. So the advantage is you're not using copper. It breaks up the algae. It's also supposed to break up the cyanotoxins and taste and odor compounds, where the copper won't do that. Again, the disadvantage is Green Clean tends to be more expensive. But we're going to be doing a treatment off of Cat Beach. As a matter of fact, from what we hear, it's getting pretty green out there, and I've just been corresponding with our treatment crew to get out there so we can collect the data and then conduct the treatment. So we already have the permit to do it. We just have to schedule that. And we were waiting for a time where it was getting a little funky, and just from some of the emails we've been getting, it sounds like it's there. So we'll be implementing that project very shortly. We're also installing biochar. So biochar is this processed wood material. The nice thing about biochar is it's cheap. And another nice thing about it is it's basically processed wood. So once you're done with it, you can actually use it as mulch. And so we had a training session with all 4 municipalities, their Department of Public Works. I wasn't at the training session, but the project manager, JP, was there. And from what I understand, it was a very positive meeting. Everyone was very excited about it because, number 1, the stuff is cheap. And number 2, when you're done with it, you can just use it as mulch. And that was the biggest thing. A lot of the filter media that we've used in the past, there's been issues with how do you dispose of it. This stuff you can literally use as mulch. And it's good for anywhere between, they say 3 to 6 months. I'm sort of hoping we can get maybe 6 months out of it, because I think it would be really nice if the municipalities would install this stuff, say in May. So you go through the whole growing season, and then September, October, fall rolls around, you can remove the stuff and put new stuff in either that time or in early spring, so that you're basically removing that phosphorus during the growing season when people are recreating. So we've implemented a set in some streams, and we've gotten some decent removal efficiencies. We've also just installed some in some local ponds that drain into the lake, as well as coves. We have to sample those to see how they're doing. So the biochar installation is something that we're looking forward to, and because eventually what we'd like to do is have the municipalities start doing this on their own. That's why we had the training session. We're also working with a subcontractor to clean out those. We have 2 large aquafilter units, some stormwater basins that were installed from one of the previous grants. We're looking to clean that out, again, of the older expensive filter material. We'd like to try the biochar in there instead, since it tends to be a lot cheaper and you don't have to dispose of it in a special manner. So we're working with a subcontractor to get them out there to clean those structures out and we'll fill them up with biochar to see if we can enhance the phosphorus removal rate. The Commission and Foundation has also been working with Rutgers on the rain garden program. So we're not directly associated with that, but I know the Commission and Foundation have been working with their rain garden program. I've seen some phenomenal stuff that Rutgers has done in some other watersheds. So that's very exciting, because those rain gardens, while you think of it, it's not treating acres and acres of land. They have high phosphorus removal rates. So if you're concerned about your piece of property and the phosphorus your piece of property generates, having a rain garden, the removal efficiencies can be high as 80%. So you think of a community where a large portion of them have those rain gardens, and you can tackle a decent amount of that phosphorus. And then finally, I do want to say that this $500,000 grant is also supported with monetary and in-kind match of an additional about $300,000. And this additional money and in-kind match was provided by the Commission, the Foundation, the municipalities, and Rutgers. And I should have mentioned also the counties contributed to that as well. So there was a lot of buy-in with a variety of stakeholders in this project. And then I do want to mention that the Borough of Hopatcong received separate funding to implement an aeration project in the River Styx Crescent Cove section. Now, we were out sampling the day before the system went on. I'm hearing good things. I'm hearing that in the center of River Styx and Crescent Cove, you're getting a nice clear channel. There's still quite a bit of weeds along the edges, but people are saying there's a positive benefit. And again, I want to emphasize, mixing the water, keeping it oxygenated, it really makes it more difficult for those blooms to form and to move up and down the water column and accumulate along the shoreline. So again, think of the three things that will really stimulate a blue-green or a cyanobacteria algal bloom. It's high phosphorus concentrations, warmer water temperatures, and still water conditions. So I'm going to wrap up here. This is just showing you what the phospholock treatment and landing channel look like. So the photo over to the right is we actually collected that with our drone. We got to use our drone for that, and then that's showing you that treatment that was done in landing. And this is showing our crew out there installing the biochar. So they go into what they call these sausages. You install them, and then, like I said, we've done one stormwater sampling event. We have at least one more to go, but then we also have to do some monitoring in the ponds. The streams, we were getting maybe 50–60% removal rate, which isn't surprising because it's flowing water. The ponds, we're supposed to get even higher because that's still water. So we're really looking forward to sampling the ponds to see what we get. But again, this material is very cheap. It's not that expensive. And when you're done with it, you can use it as mulch. So that's why and I gotta say, all 4 municipalities, when we mentioned we'll be out there, all of them wanted to participate. They wanted to see what we're doing. They're really hoping this is gonna be something that they can do to contribute. It's not gonna cost an arm and a leg, but it's gonna have a positive benefit for everyone at the lake. So to wrap up, so moving forward, we're gonna evaluate the relative effectiveness of all these nearshore HAB projects. We want to finalize the WIP, but then we want to go into the design, permit, and implementation phase. So we'll be looking for funding from a variety of sources for stormwater watershed projects. Also the sewering of the watershed. So, the Borough of Hopatcong and the state park are doing some sewering. Jefferson is updating their sewer plan, and this is good because if we do want to see full compliance, there's going to be the need for a higher level of sewering. So this is a step in the right direction in terms of full compliance. So speaking of full compliance with the TMDL, our long-term goal is to get the average concentration throughout the lake at 0.03 milligrams per liter or less, and to have chlorophyll A at 8 micrograms per liter or less. And then finally, we're going to evaluate the monthly and seasonal role of internal phosphorus loading from the sediments. I can tell you that internal loading on an annual basis for Lake Hopatcong is only about 7%. So it's less than 10%. But what we're wondering is during the summer or during certain months, as the blue-greens can move up and down the water column, case in point of last year, even though the phosphorus concentrations in the surface went down, the blue-greens persisted in Lake Hopatcong. Other lakes we work on, like Harvey's Lake, they disappeared. they stayed at Lake Hopatcong for the whole year. Why is that? We think it might be because of internal phosphorus loading. So we're hoping to evaluate that. The commission submitted for a 319 grant, which would implement some stormwater projects, but we would also do this analysis of the monthly and seasonal role of internal phosphorus. And fortunately, because we have a 30-year database, we have a lot of data we already need to do that. So it's not like we have to go out and collect a ton of water quality data to do that analysis. We have a 30-year database that we're going to rely on. Again, another value of that water quality data. So I know that was a lot of information to throw out at you, but thank you for your time. I really appreciate it. And if you have any questions, feel free to ask.
Hi, Fred, it's Donna. So if you wouldn't mind just taking a minute to unshare your screen, and then we will go to some question and answers. But before we do that, I did notice in the chat that there were a couple questions regarding what homeowners or people that live within the watershed, what they can do to help improve the lake water quality. And I just wanted to say that is a perfect lead in. Our next Lunch and Learn webinar will be on August 20th, where Jess Murphy, our president, and myself will be talking about lake-friendly living and what homeowners can actually do. So as we go through the questions, I'll ask Dr. Lubnow in the interest of time, because we have about 10 minutes left, to kind of briefly touch upon some of those questions. But I really strongly encourage everyone to join us again on August 20th for that lake-friendly webinar one, where we're going to be addressing those more in depth. Okay, so if we go to our chat screen now, we have a few questions, and the first one is from Robert, and he's asking about what can homeowners do besides using phosphorus-free fertilizers, and can they continually pulling the floating garbage out of the lake and moving weeds. So I think you talked about that a little bit relative to the rain gardens, but do you want to briefly touch upon that as well, Fred?
Sure, absolutely. So if you have a pet, pick up after your pet, so the pet waste doesn't wash in. Anywhere, if you have shoreline property and anywhere you don't have like a dock or a beach, try to have some vegetation growing, maybe about as high as your knee. You can use some attractive native vegetation, or you can just let it grow as high as your knee. That's really important for a couple reasons. Number one, when you have that nice buffer of native vegetation along the shoreline, that helps to remove nutrients that otherwise would go and fuel algae growth. But it also deters geese from going onto your property. And it's sort of funny, I've seen this in Pocono communities where I'll be with a property manager, excuse me, and we're walking across a number of properties, and properties where they have vegetation about as high as your knee, you see no goose poop, and you will literally walk 10 feet over and you see someone who has a lawn right to the edge and you see goose poop all over. Geese produce a lot of phosphorus. So, having some sort of shoreline vegetation, if you're on septic, and Jefferson's pretty good about this, pump your septic system out at least once every 3 years. That alone, you'll reduce your septic-related phosphorus load by anywhere between 10 to 30% just by pumping out once every 3 years. So picking up after your pets, trying to have as much green space on your property as possible. And when I say green, I don't mean just like lawn, but native shrubs and plants and vegetation like that. Pick up after your pets. I mean, all that really comes into play. And then the rain gardens. If you can design a rain garden to take a lot of the runoff from your property and put it into that rain garden for treatment, that can be very useful as well.
Thank you, Fred. So I think we addressed Robert's question relative to the geese poop when we were talking about the importance of shoreline buffers. I myself can attest to that. We have buffers along my property and my neighbor's property, and a new homeowner came in and took down the buffer along his shoreline, and then the geese did not hesitate to come up. And because we have no fences between our yards, they felt free to walk all across. So the neighbor did eventually understand the importance of buffers along the shoreline. It really does help to keep the geese off the property. We have a question from Norm about asking if cyanotoxins are they problematic to humans and pets, and can we directly measure these? Why not rely on these measurements instead of cyanobacteria counts, which may or may not represent a problem when it comes down to limiting usage of the lake? Kind of a long question, but can you give us a short response, Fred?
No, but you know what? It's a good question. And one of the reasons why we don't focus on directly monitoring for cyanotoxins is there are dozens upon dozens of cyanotoxins. So using the cell count is more of a safe way that, okay, we're only sampling for 3, but there could be a rare dermatoxin or something like that being generated. You would have to sample dozens upon dozens of them. Last year, we had a lake community who insisted they wanted a dermatoxin analyzed. We analyzed it for them, but that one sample was $250. When we measure it in the field, we're doing like essentially presence absence. So it is a quick test, but it's not giving you a number. And the potable water people and the state agencies, if you're going to test for that, and they want to know what that concentration is, you have to send it to a lab. And unfortunately, when you send it to a lab, the quickest turnaround is maybe 4 days, 5 days, and that's quick. So by that time, the bloom could be worse. The bloom could go away because of prevailing winds. So that's why we use the meters. So we do this little test kit. We'll do the test kit, and it at least gives you a presence absence, so that we can provide that information. But the cell count is a definitive way of saying, look, there could be a potential for problems here of something we're not even measuring. So that's why they hang their hat on cell counts. But even cell counts can take a couple days to get. So what DEP is doing, they're actually recommending eventually using a meter for measuring phycocyanin. Phycocyanin is a pigment that only the cyanobacteria generate. And so we've purchased phycocyanin meters, Princeton Hydro, and as part of the HAB grant, every time we do a cell count, we're using the meter. The goal is we want to generate a relationship between phycocyanin and cell counts for Lake Hopatcong. And this has to be done lake specific. But the value to that is then if you have a calibrated meter, all you have to do is you can go out and measure the phycocyanin and then make a determination. So the nice thing about that is, when we get a cell count, it takes us at least 48 hours to do it, because once you have the cells, you have to let the cells settle, you have to concentrate it. It takes at least 2 days to get that data together. The phycocyanin, anyone could do it. You read it, and you have that data right away. So what we're hoping is eventually we'll have this relationship. So if a beach community wanted to invest in one of these meters, they could use this meter to make a determination of at least providing guidance on what the water may be like for potential cyanotoxin risks.
Thank you, Fred. I'll just add, the Watershed Institute is hosting a free webinar later this month on August 26th, where folks from the DEP will be talking more in depth about this and how people can get involved in voluntary monitoring of HABs that the DEP is trying to implement. So I will send that information out to everyone who registered, and if you're interested, you can register for that directly through the Watershed Institute. We have just a couple more minutes, Fred, but I want to just try to get to this one about the biochar. This person is asking if it could be used for a failed drain on her street that's bringing everything into Byram Cove, and can we apply for our properties? So just to clarify, the biochar is not a rebate program like the rain garden one is, where people, if they install the rain garden, they could get a rebate up to $450 if it was designed by Rutgers or approved by Rutgers for that implementation. The biochar, Fred, can you just talk a little bit about how Princeton Hydro selected these locations this is really an evaluation of this new media that we're trying to implement.
Yep. Yeah. You hit it right on the head, Donna, that it's been around for a while, but it's relatively new for lake management. So we are evaluating it, and they do have different size of particles. We were very careful to select sites where we felt we wouldn't cause flooding. And that's the one thing, if you're installing it in some sort of street drain or anything like that, you'd have to make sure the structure has the capacity to hold that. So I mentioned the aqua filters, those basins that we're going to install biochar. They have a separate compartment for material like that. The thing I would be concerned about is if you just put it in a catch basin, If you have a large enough storm, that sausage may result in clogging a pipe up or getting stuck somewhere and causing some local flooding. So my answer is you could use it, but you would need to work with your local public works department and the municipal engineer to make sure that it's safely installed so it doesn't cause any localized flooding.
Thank you, Fred. And Kerry Green is asking regarding permitting. Do the towns need any permits through DEP to install the biochar filters on the streams?
Yeah, we actually went through that, and specifically for Lake Hopatcong, we did not need any permits. It was permit by rule. But we're working on a project with New Jersey Water Supply Authority, and they're putting it in one of the main inflows going into Spruce Run. There, I think we're going to need a permit. So I think it's going to be site specific, and it really depends on the size of the water body and waterway. So we were putting them in small streams, very localized areas at Hopatcong, or we're putting them in ponds. So we have some of them in Duck Pond, some in Memorial Pond. So there aren't any issues relative to permitting, but we did go through that, and it was determined permit by rule. We're okay with Hopatcong, but I do know for a Spruce Run, we're going to have to go through some sort of permit process.
Okay, thank you, Fred. And we're going to close out with one quick question from Jim Fitzpatrick about asking about the current status of HABs and the prediction. I know you don't have a crystal ball. We all wish we had one, that we could tell what the future would be for so many reasons this year. But
Yeah.
Can you just tell us a little bit about the status of HABs for this year?
Yeah, this year is looking more like a quote normal year unquote, in that we had a decent June. We're going into we went into July. We're going into August. It's starting to get a little funky out there, and we're supposed to get some storms. But when I look at those high temperatures, I'm really concerned about the development of HABs. We're already seeing some of the shoreline areas are starting to have those nasty HABs, and we're already going to go out and do some of these treatments. This is more of what we typically see at Hopatcong. Now, the best case scenario is it would be nice if it waited until after Labor Day, but unfortunately we don't have that luxury. But the way it's going, we may be struggling with some HABs this month before we get into Labor Day.
Well, thank you, Fred. I hope everyone will please just join me to give Fred some virtual applause here. We really appreciate him sharing his time and his extensive knowledge with us today. He's really a great partner for the foundation and our lake community. So thank you, Dr. Lubnow. Everyone have a wonderful day and be safe. Thank you.
Thank you, everyone. I appreciate it. Thanks. Take care.